Good morning. I am your host, Facundo Batista. Together with Richard Young and Canum Overhaul, we have organized this MIT course on COVID-19, SARS-CoV-2, and the pandemic. On behalf of the three of us, thank you for joining today. The purpose of this course is to learn what we know about this virus and the pandemic from the best scientists across the world. It is my privilege to introduce to you today Professor Andrew Warts, an incredible talented young scientist and a fantastic colleague. Andrew grew up not far away from here in Melrose, Massachusetts, and he obtained his Bachelor in Science at Duke University. And very early in his career, he moved to Scripps to learn cryo-EM microscopy that was part of his PhD studies. His postdoctoral work focused in structural biology and biophysics of membrane protein, and in 2012, he established his independent lab and began part of the faculty at Scripps, where he's now full professor. One of the primary focus of Andrew's lab is to understand how to improve antibody-based therapies and rational vaccine design. In my view, Andrew is and was a scientific visionary. Very early in his career, he understood the transformational power of cryo-electron microscopy can offer to solve these problems. And indeed, by doing so, he was the first to solve the structure of unstabilized HIV capsid. That is a fundamental step towards the development of a vaccine to HIV. But his achievement didn't stop there, and his vision either. Very early, he saw the power of this technology and solved, back in 2016, the first structure of coronavirus spike, which led to the creation of a stabilized version of the protein, which is now used in practically all the vaccines that are on the market, such as Moderna, Pfizer, J&J, and Novavax. Andrew was awarded the Wright Thomas Edward Foundation Fellowship. In 2014, he was recognized by the Palmer Jr. Investigator Award by the American Society of Virology, and in 2017, was a recipient of the Virus Young Investigator Award in Virology. In my view, this is just the beginning of what is already an incredible career. And Andrew, thank you very much for joining us today. It's very early for you, and we're really looking forward to your talk.
Thank you, Facundo, for the very, very kind introduction. It's actually, a pleasure to be a part of this lecture series with quite the lineup of speakers. And so, as Facundo mentioned, this is sort of a virtual homecoming for me. I wish I could be visiting Boston in person to visit family and friends, but I'm doing it remotely from San Diego. So I'm going to really give you a few examples today of how structural biology has played a role leading up to the coronavirus pandemic, as well as currently. So really describing structural approaches to combat COVID-19. So in this very short amount of time, I'm going to do the ambitious task of covering a quick introduction to structural biology, which I hope is an inspiration to many of you as undergrads, just like it was to me when I first learned structural biology, and then go in and describe how structure-based vaccine design is undertaken, and then a little bit about antibody responses to SARS-CoV-2 and how that shapes what we do for vaccine design and immunotherapeutics. And then I'll end with understanding some structures of SARS-CoV-2 proteins and how they can be targeted with structure-based vaccine design. And really, structure provides, as I'll show, examples of logical puzzle solving that become quite intuitive once you get past the initial complexity. Okay, so why structure? I particularly like this quote from Max Perutz, a former Nobel laureate that solved the first X-ray crystal structure of a protein, myoglobin. And he stated, why water boils at 100 degrees Celsius and methane at minus 161 Celsius? Why blood is red and grass is green? Why diamond is hard and wax is soft? Why graphite writes on paper and silk is strong? Why glaciers flow and iron gets hard when you hammer it? How muscles contract, how sunlight makes plants grow, and how living organisms have been able to evolve into ever complex forms. The answers to all of these problems have come from structural analysis. And likewise, as I'll show, structure plays quite a role in combating pathogens such as SARS-CoV-2. So here's the textbook version of the viral life cycle of SARS-CoV-2. So the first step is that the virus must enter the cell. It does so by using these spike proteins, which you'll hear a lot about, interacting with the ACE2 receptor and entering the cell. And so outside of the cell, this is the target for antibodies. If antibodies can block this interaction, then of course the virus never gets in, it gets cleared, and you have immunity. So we're going to talk about vaccines, particularly to the spike protein and inhibiting this process. Once the virus gets into the cell, it starts making proteins and making copies of itself in progeny virus for release. So there's a lot of different proteins doing a lot of different functions, of course. And if you can inhibit one or more of these, then you have a chance to slow down the virus. And of course, antibodies don't cross into the cell. So now we're in the small molecule regime. And so one of the first or one of the most popular targets is inhibition of this viral protease and processing of these polyproteins. And I'll show an example of that. And then the other is in the RNA replication and transcription. So the virus needs to make new copies of its genome for packaging, as well as RNA transcripts to make more viral proteins to infect the cell and proceed. So that's what the textbook description of your cartoon description looks like. But of course, there's a lot more detail that we can understand by investigating the individual structures therein. So here's just a really beautiful diagram. This was part of a challenge that I was a judge for a software called AutoPack, wherein atomic models solved by structural biology are used to populate realistic regimes. And so this is showing an infected CD4 positive T cell. So this is an HIV infection. As HIV buds out of the host cell, it packages its genome into this coffin-shaped capsid. The virus is wrapped in this envelope derived from the host cell membrane. And you can see these red spike proteins on the surface, as well as some other proteins. If this virus goes undetected and detects and recognizes another CD4 positive T cell, it can start the process all over again, and that's how infection spreads. But there are these blue things. These are the antibodies. If these antibodies recognize the virus, they can bind to, prevent recognition of host cell, and eventually lead to clearance. So this is really just a wonderful description that's correct at the molecular atomic level that really paints a much more complex picture than that diagram I just showed. So here is the sort of working inventory of proteins that are encoded by the SARS-CoV-2 genome. So it's 29 different proteins, each with a specific function as part of the viral life cycle. And what you can notice is that the first thing is they're different sizes and they're different shapes. And so really, as I'll show, the shape encodes the function. And in order to get these structures, it's a combination of experimentally derived structures here in this peach colored line, and then there are modeled structures. So homology model based on similar structures, or de novo model based on AlphaFold, which I won't talk about, but a lot of the machine learning structure prediction tools are quite exciting and emergent. So today, as I mentioned, I'm going to focus on the spike protein, which we've done a lot of work on and is the target of vaccines. Talk about the protease, as well as the replication machinery over here. All of these other proteins are potential targets for different parts of the life cycle, but these are the three primary targets for vaccines and small molecule drugs. So in order to really talk about proteins, I need to step back, and hopefully many people have heard of the central dogma in biology. And that is that your genome is encoded in DNA. So these are the master blueprints in the nucleus of your cell that are carried forward and encode the 20,000 or so genes that make you a human being. This DNA is transcribed into RNA or messenger RNA. And these are the transcripts that go into the cell and then are encoded into these proteins. And so this is the flow of information. The proteins do all the work, and you have the master blueprints, and the RNA is the messenger in between those two. Viruses can have a DNA genome or an RNA genome. Either way, they're encoding a set of proteins. And now, as most of you are famously aware of the vaccines that are being developed, you can introduce messenger RNA, specialized messenger RNA into cells, into the body to make certain proteins. And in this case, the spike protein of coronavirus, and that protein will then elicit an immune response. So a little bit of chemistry, just a little bit, I promise. So proteins are polymers made up of these individual amino acids, and they form polymers known as polypeptides that fold into three-dimensional structures. So there's a common backbone that are encoded by all of the amino acids, and then each one has a different side chain here depicted in all these different colors. And each of the side chains is a different shape, but also has different character. So some are sticky, some are greasy, some attract other amino acids, some repel other amino acids, some can form covalent bonds. And so based on these different building materials and combinations of building materials in this polymer, you get folding into different structures. Structures, again, dictate the function of the protein. So here's a nice little YouTube video showing really how this works, a really simplified—
Proteins are building blocks that help.
Mute that. So here's just, you can see a linear string of polypeptides. Lots of different amino acids are encoded in this video, and you can see that these can be represented by these single letters, or they can be represented as beads on a string. Again, each one of these has a different character. When these proteins are made in the cell, depending on the sequence, they will spontaneously fold from these extended polymers into three-dimensional objects. In these three-dimensional objects form, you have local structure like alpha helices, and then you have long range interactions. And so you can see that from this basic set of 20 amino acids, you get this folded functional shape. And by understanding and knowing the structure, you can also manipulate and change some of these amino acids and either change the structure, change the function, or create vaccines, as I'll show in a minute. Okay, so how do we get there? So I mentioned through Max Perutz the power of X-ray crystallography, which is about 100 years old. Something that's much newer is cryo-EM, and that's really where my lab has focused its attention, as Facundo alluded to. And as a graduate student, before cryo-EM was really at the forefront and capable of doing what it could do, I really appreciated this quote from Richard Feynman, from a lecture from 1959. And he said, it's very easy to answer many of these fundamental biological questions. You just look at the thing. Make a microscope 100 times more powerful, and many of the problems of biology would be made very much easier. I exaggerate, of course, but the biologists would surely be very thankful to you, and they would prefer that to the criticism that they should use more mathematics. And that's exactly what happened during my early career, is that the microscope was made really 100 times more powerful. And so this was actually recognized, cryo-electron microscopy, as a 2017 Nobel Prize by recipients Jacques Dubochet, Joachim Frank, and Richard Henderson. And here's what a modern electron microscope looks like. And in addition to the innovations made by by these three gentlemen. There's also these amazing cameras. So it's really analogous to how your iPhone camera uses live imaging. So, you can take a live photo, and if you try to capture a moving object like a toddler, you can actually align those images and get a much crisper picture. And that's really what these cameras are doing. So you have the high-end optics of this electron microscope coupled with these exquisitely sensitive detectors or cameras, which yield atomic level images. And just to give you a sense of size, even though this is small here, this is actually, the size. These are about 10 feet tall. Here's a picture of my daughter, who was 2 years old at the time, standing in front of all of this complicated set of wiring and technology. This is where we actually put the samples in, and you can see that even though this is a simple box, it's really a $5 to $7 million box. So these are very expensive toys, but they can do quite exquisite things. Okay, so this is sort of, the wonders and really inspiration, right? This is an HIV antibody on the left, reaching out and interacting and neutralizing the HIV virion. And just to really show you the beauty and, again, intuitive nature of structural biology, you can see that each of the different amino acid side chains has different shapes. These shapes fit together, and you have these antibodies encode these regions that can reach out and reach into nooks and crannies and bind to and neutralize viruses. And so really, this is one of the inspirations of structural biology, especially if you like fitting puzzles and looking at different shapes and forms. Okay, so using the electron microscope, my lab, as Facundo mentioned, solved the first structure of the HIV envelope-like protein. We've done a lot of work on Ebola, surface protein, malaria, influenza, hemagglutinin, and neuraminidase, as well as some early work on coronaviruses. And so, all of these structures were solved in the matter of a few years. And this normally, before cryo-EM, would have taken a lifetime or beyond to solve these. So really just showing you how we can turn the crank now and look at a lot of different problems and use these structures for, in this case, vaccine design. Okay, so, as the cryo-EM community was small at the time, we all appreciated this and its potential. The rest of the world slowly caught on. And the NIH Directors Blog in 2014, Francis Collins, the head of the NIH, had a really nice article that featured our use of electron microscopy to image ZMapp. And ZMapp were these 3 monoclonal antibodies that bind and neutralize Ebola virus and were used during that outbreak. And you can see here in red, blue, and yellow binding to the surface spike protein on Ebola. And then really, gratifyingly in 2016, in Francis Collins' address to Congress to justify the NIH budget, he cited a single scientific study, which he called a compelling current example of how basic science is revolutionizing the future of biomedical research, is in the potential treatment of deadly Ebola virus, and went on to say, it's important to know where those antibodies bind to the protein and how that would interfere with Ebola's ability to infect cells. So again, really demonstrating the impact and power of visualization and understanding at the structural level that cryo-EM was enabling. Okay, so flash forward to 2020. This is one really beautiful example of a use of an electron microscope from John Briggs's group at the MRC early in the pandemic. He was imaging SARS-CoV-2 virions, and here's just showing 2 different virions reconstructed. There's different numbers of these spike proteins on the surface. These spike proteins can twist and dance around, and really, again, this was a Herculean task that was quite undertaken quite quickly once the pandemic started. And so that viral spike protein is really the key linchpin to getting into the host cells, as I showed. And so here's just a really beautiful video showing, how that happens. So you have the spike protein on the surface. It has the S1 receptor binding domain, and then the yellow fusion machinery. Once an ACE2 expressing cell is recognized, S1 dissociates, and then you have this incredible conformational change. So that single protein that's folded up starts to refold and undergo this set of transformations that ultimately bring the virion and the virion membrane close to the host cell in order for the host cell to become infected. And so if you can target this process, you can stop the infection. So, again, just really showing how remarkable these proteins are at rearranging and shape shifting. Okay, so that brings us to rational vaccine design. So because these proteins undergo those transformations, they're inherently unstable. And so we need to do a lot of engineering to stabilize these surface proteins. That's done by solving structures and then going into the computer and making changes and modeling how replacement of certain amino acids would have a different impact. Once we have a design, this can be delivered in different ways. So you all know about the RNA vaccines. They can be delivered as a message. They can also be made as recombinant proteins, such as Novavax, which actually makes the protein in a bioreactor and delivers that. So it cuts out the step of people making this with their own cells. J&J and others are using vectored vaccines, which again are a viral vector to introduce this spike protein. And then there's the more classical approaches of inactivated vaccines, which deliver a weakened form of the variant. But in all cases, the spike protein is the main antigenic component that we're trying to elicit immune responses to. And so in the sort of iterative part of this process, these need to be tested preclinically in animals and eventually in human clinical trials. We have classic serology and immunology that's done, but increasingly we're using tools like next generation sequencing and structure to really inform the redesign of this process. So there's a lot of rational design and then empirical testing, and this can happen quite rapidly now by many of the tools that we have. So the concept of structure-based stabilization of these metastable viral glycoproteins is about 20 years old. And the details don't matter. Here's just showing the HIV envelope glycoprotein trimer, which was really a work in progress for 30 years. And it took these protein engineering approaches. RSVF is another example where Jason McClellan and Barney Graham and colleagues were trying to stabilize the respiratory syncytial virus in this pre-fusion conformation, and using these tricks like adding disulfide bonds or molecular staples for RSVF, or adding prolines to stabilize this pre-fusion conformation and prevent those extensive conformational transitions were critical for HIV envelope. And really, this is key because without stabilization, you elicit the wrong type of antibody responses. So we knew from these basic examples, how critical that was. And my lab is working with many colleagues to do structure-based design here. And as I mentioned, Jason and Barney doing here. John Moore and Rahir Sanders had a really nice review recently entitled Virus Vaccines, Proteins Prefer Prolines. And these turn out to be pretty magical amino acids that you can introduce to stabilize proteins. So 2015, that, as I mentioned, Jason and I were working independently on different projects. 2015, he reached out, and in the backdrop of a small-ish SARS outbreak and MERS outbreak, we thought to join forces and work on coronaviruses. I had a postdoc at the time, Rob Kirkdorfer, who was interested in learning cryo-EM, this emergent technique, and so it all sort of coalesced And, over the course of a couple emails, we decided to join forces and fortuitously work on coronaviruses. Jason was going to send us some of the proteins he was trying to make, which were in minuscule amounts. He was going to show him, some things about cryo-EM, and we we're going to divide and conquer and do this thing and use the lessons learned from HIV and RSV. And as I said here, hopefully we'll be successful, publish lots of papers, Rob gets a faculty position, and we'll keep collaborating and meeting up at scientific meetings for beers. And really, this was the beginning of a wonderful collaboration and lots of scientific success. So we were interested in MERS and SARS spike proteins because those were the pandemic ones. We couldn't get those. They were unstable. We couldn't make them biochemically. They were challenging. But we happened upon a related virus, HKU1, which causes seasonal colds. And we were able to solve it. Again, cryo-EM was critical to this because we couldn't make that much of the protein. But once we had the protein, it went quickly. We solved the high resolution structure. Again, you can see here's the shape it adopts, the receptor binding domains, the fusion machinery. And from here, we said, okay, we can now use this as a template to do structure-based design and add those interesting mutations to try to stabilize the other versions, the MERS and the SARS spikes. And so this was well before the pandemic in 2016. So we were working on MERS at the time. Here's just a spreadsheet showing you, that even though we're rational and structural biologists, we still need to test lots of different mutations. Many of them don't work, some do, but this is an iterative process. But of course, having the structure reduces the amount of time that's necessary, right? Because there's 1,300 residues and there's 20 amino acids. You can do the math. If we were to just saturate and do all of those mutations, it would be in the tens of thousands and take forever. So again, taking a page out of HIV stabilization, we knew that if you add prolines into the fusion machinery in particular regions, here showing in this central region, in that movie, I showed that this helix undergoes a very large conformational change into this extended form. If we added one or even better, two prolines to this region, it prevented this conformational change and opened up the door to producing the MERS and SARS spike proteins. So what you can see here is that when we were in the wild type sequence of the virus, we were making almost minuscule amounts, but when we added these mutations, we got 50 to 100-fold increase in expression. And then looking at the electron microscopy images, we went from a distribution of these post-fusion or undesirable forms of the protein in the wild type sequences to entirely these pre-fusion and desirable shapes. We went on to show that these 2p mutations were universal. So for any beta coronavirus that we added these two, we got stabilization, expression, we could solve structures. And so this was now a universal platform that could be used and kind of was there and on the shelf until the pandemic. And so once the pandemic hit, from the published and patent literature, people rapidly incorporated these 2p mutations into SARS-CoV-2 spike. And as Gunda mentioned, Moderna and Pfizer, BioNTech, Johnson & Johnson, Novavax have all incorporated these 2p mutations. And so it has the advantage, as I just showed, of an increase in the expression level. So that's good for immunogenicity, as well as a increase in immunogenicity. So really, this is just a nice example of how years of basic research enabled the vaccine development. So the 2p mutations, and then of course there's a whole history of mRNA development that came to fruition during this pandemic. And so working in a vacuum well before a pandemic actually pays off and allows us to do things quite rapidly. So now the goal is to really to look at where these antibodies that are listed bind on the spike protein. We can compare CoV-2 infection to mRNA vaccination. The different antibodies neutralize in different ways by acting on the spike protein. And this is sort of a summary. We know that antibody responses target particular regions, either the NTD or the receptor binding domain. The receptor binding domain is really the critical piece that you want to block. Within the receptor binding domain, there's been a lot of structural work showing how these different antibodies recognize. You have different classes that bind different regions. Regeneron, the immunotherapeutic cocktail that Regeneron made, utilized these two different types of antibodies, and there's other immunotherapeutics coming along the way. We know now from structural studies that the class 1 and 2 block that ACE2 receptor engagement. They're highly potent, but they're susceptible to variants of concern and mutations. 3 and 4 are less potent, but also less susceptible to variants of concern. Class 4 is interesting because you can get cross-reactivity to other SARS viruses. And so there might be potential for more universal SARS vaccines by targeting that region. So you're just, showing there's variants of concern plotted onto the receptor binding domain, as you might have heard. Many of these actually improve the interaction with ACE2 receptors. That's an advantage for the virus for getting in the cell and the pathogenesis. Here's just some examples of antibodies. And so there's a lot of work being done to identify antibodies that don't interact with these variants of concern and therefore are not susceptible to the virus mutating. And again, structural biology playing a big role in identifying the molecular details of these interactions and how what makes a good antibody from a bad antibody, what makes an antibody likely to avoid a variant of concern. And then there's a lot of work being done, again, using protein design to develop new versions of the vaccine that are only, for instance, the receptor binding domain. We now know that small piece is really critical for neutralization. However, if you immunize with just that small RBD, you get a weak response in your B cells. If you use protein design like Neil King and David Baker do at the University of Washington, you can build these nanoparticles that have multiple copies of the receptor binding domain. You get a much more potent B cell response. And again, showing you in the matter of a couple of years, using all of this structural information to make next gen and next gen vaccines. So we've gone from understanding the structure of the spike, stabilized version, where antibodies bind, to really designing now booster vaccines, smaller subunit vaccines, as I just described, and pan-coronavirus vaccines. And really that's, a quick journey through the work that's been done in the coronavirus space in a really remarkable amount of time. And based on our ability to rapidly iterate, so take advantage of pre-existing knowledge and then rapidly iterate through structural biology, immunology, and other technologies that I'm not mentioning here to really produce what are remarkable vaccines and what will certainly change the landscape of vaccines for future pathogens, viruses, bacteria, and otherwise. So that I'm going to transition now to a non-immunology approach to target the virus and highlight the aspects of structural biology. So we just talked about blocking the recognition of host cells by those antibodies. Now we're going to move into the other targets, the viral protease, as well as the RNA replication machinery. And this is a really nice review from Yang and Rao that came out and again highlights where a lot of the effort in academia and industry is at stopping the virus. So here's our protein inventory again of different structures. I'm going to talk about these RNA-dependent RNA polymerases for which we had some pre-existing structural knowledge and a lot more now that the pandemic has happened. And our work in my lab began again with Rob Kirkdorfer when he was a postdoc, and again, cryo-EM played a critical role in being able to solve the original structure of the SARS-CoV polymerase. And so, as you can appreciate, this has its own shape, and it has this beautiful channel here through which RNA is threaded, and just showing the different views. And once we had this structure, this, of course, set in motion the ability to think about how to target this with small molecules and drugs and inhibit the function. There was also some knowledge of other viral polymerases, such as from poliovirus or dengue virus. And while the details are different, you can see that the overall shape, really exemplified here by the poliovirus, is similar. So these are all related and evolved from one another, but elaborated in different ways to have different functions, or the same function, but to do slightly different things for the viruses. Okay, so how do these polymerases work? We know a lot about polymerase replication, not only from the work on viral polymerases, but humans, of course, have polymerases too. And what happens is you have a template strand, and you need to make a copy of this strand. And this is what this enzyme does. And it does through this base pairing. So these nucleotides are floating around. It picks up a nucleoside, and then it finds the right match. So for every blue base here, it adds a green base. If it does that productively, it moves on to the next one, and so on and so forth. And you get replication of the daughter strand. The way something like remdesivir works, and remdesivir was, of course, being developed as an anti-Ebola drug or an antiviral drug in general. But because of the similarity of the polymerases, it was tested early on to see if it had an impact on the function of SARS-CoV-2 polymerase. And it did. And so you can see here what happens is it looks like one of the green bases here, but it has a small modification, as I'll show in a minute, that gets recognized and incorporated, but it causes the polymerase to fall off and stall. So you block the replication. There are other inhibitors like favipiravir, I think is how you say it, in which, again, small modifications lead to incorporation. In this case, the polymerase doesn't fall off. It continues to extend, but it makes this daughter strand that's completely non-functional. So again, it's using tricks to incorporate the wrong type of bases into this template strand, or into the daughter strand. As I mentioned, early work was done on the SARS-CoV-2 polymerase or other viral polymerases. There's been a lot of recent work. Here's a structure from Patrick Kramer's lab, really showing the details of SARS-CoV-2 polymerase. Caught in the act, I really like this image and showing how remdesivir fits in. And so we, of course, have a lot of molecular details and what people are interested in. Again, zooming in, the details don't matter here, but what people do look at are the molecular interactions, and also look at where viruses may mutate to then overcome this drug. So viruses are really good at mutating, and if you put a lot of pressure on them by adding drugs, they often can escape that drug. And so by understanding the structure, again, at the atomic level, we can design drugs that potentially can't be escaped from. Okay, so I mentioned, how we're using structure-based vaccine design. Here's what the correct nucleoside looks like. ATP, remdesivir, only has this small elaboration, and this elaboration is enough to cause the polymerase to fall apart. And so, when you zoom down to the molecular level, this is really just optimization of shape, right? So when you're designing these drugs, it must be similar enough to the native substrate to fool the polymerase to want to take it out, but it must be different enough to have an effect. It must be specific enough to prevent off-target toxicity. So there are human polymerases, and so you have to target specifically. And this is a big challenge of small molecules, is the off-target responses. But once you have the structures known, you can really rationally design through different elaborations. And it's really just, this process of when you understand, such as this child's toy, how different shapes and objects fit in, you can add elaborations to them that prevent them from being fully functional. Okay, so I'm going to give a last quick example of what's people are doing in terms of the protease. And so, as I mentioned, this is the processing of the polyproteins that are made. So when the virus gets into the cell, it's making these polyproteins, which are going to do jobs for the virus and help replicate. And what it does is, as these polyproteins are made, they must be clipped into functional units. So each one of these is a different protein that does a different job, but they're only functional when they're clipped. And so this has been under intense study because it's known from other viruses such as HIV. Protease inhibitors are a good target to slow down the viral infection. And so here's just showing you different inhibitors of the main protease. And just to show you just the molecular ways in which these different shapes, these different combinations of atoms are designed to target these pockets on the surface of the protein. So what they do is they compete with the native substrate. So the polyprotein needs to bind and be clipped, but if you find a small molecule drug that binds there instead, you inhibit this process. And there's lots of different ways to do this. In fact, there's many, many different ways, and just showing you so you can appreciate some of the designs, how they both fit the shape of this pocket, but also the molecular interaction. So taking advantage of what's known about the side chains, you can optimize the different functional groups of these small molecules to interact with the different regions. And I would say, this is, again, been a rapidly evolving field since the beginning of the pandemic. Lessons were taken from other protease inhibitors, but encouragingly, Pfizer now has a quite advanced Mpro inhibitor that results in about 90% reduction in hospitalization when given 3 days after symptoms. So this should be available soon. And this actually uses a pretty fantastic design principle wherein it covalently modifies the protein. So the other inhibitors I showed can bind and release, and in doing that allows substrate to engage, right? So they can be good inhibitors, but they're not sort of shut down inhibitors. Here, because this covalently binds the protein, it forms a chemical bond, it stays on and therefore completely shuts down the protein. And so by doing rational design and knowing which functional chemical group to position here, you can then modify this reactive cysteine in the catalytic pocket of the Mpro. And catalytic inhibitors are really a fantastic way to shut down proteins of interest, and also a way to increase specificity and reduce off-target for human proteases. Of course, there's human proteases that would be competing. Okay, so that is the last example I'll give, and hopefully I've given some inspiration to why we do structural biology, and made many of you appreciate, albeit in a somewhat complicated sense. I know this is pretty complicated to look at, but hopefully talking you through some of these examples showed how structural biology plays a role really across vaccine design, understanding antibody responses, and developing new small molecule drugs for the inhibition of coronaviruses, as well as, of course, many other things. And most industry is using structural biology to do these type of processes. So with that, I'll just end and say, cryo-EM, we're still looking for new opportunities for drug design. In some of the cryo-EM structures we've solved, this is going back to the spike protein now, we found this small molecule, either sapenic acid or linoleic acid, and these bind these pockets. These actually stabilize and prevent conformational changes. So we're looking to exploit these now for rational design of drugs that can target the spike protein and prevent infection from occurring in the first place. So in addition to antibodies, which can bind and have an impact, there's opportunities to target the spike. And we find these opportunities, again, by looking at the structure, finding random small molecules that happen to be there, and then setting off on campaigns to develop drugs that might specifically bind there or covalently bind there, as I mentioned in the last example. So just wanted to end and really, acknowledge there's been, a huge support for a lot of my work, and of course, all the other work from different funding agencies, and I have a quite diverse and vibrant group that does a lot of different structure-based approaches for vaccine design and immunotherapeutics. If you're interested, you can check out my website or follow us on Twitter. And then I'll also end by advertising. We have a graduate program at Scripps Research. I'm actually alumni of that program, and it's a really fantastic research environment. We do a lot of great structural biology, and we are accepting applications this year into December. So if you're interested, I encourage you to check it out now, or maybe in the future when you're considering your PhD or beyond undergrad. And with that, I'll end and thank you for your attention and take questions.
Thank you, Andrew, for a wonderful talk and for making very complex scientific topics very accessible. So thank you very much for that. So there have been a couple of questions that I will ask actually. So one of them is, obviously cryo-EM has really been catalytic in the development of all this, but is that all what do you see happening in the future? What this technology is missing right now?
That's a good question. I think what the technology is missing is throughput. So there's a lot of reasons why it doesn't move as fast as it can, but those are rapidly changing. So early days, that first coronavirus spike structure that we solved, it took us a few months to solve. Now, when we solve a coronavirus spike structure, it takes 2 days. And that's a lot to do with the softwares that are being developed, as well as the imaging. So imaging, we collect terabytes of data, thousands of images. That of course takes time. But really the computational end is speeding things up, and increasingly so. Artificial intelligence, machine learning approaches are shortening that path even further. I did mention AlphaFold as a tool for protein structure prediction, and that's also playing a role. So you have technologies being combined, and I think the outlook is that we'll be able to solve the structure of any protein in the matter of days. But of course, you need to undertake the biochemistry, which can be challenging in order to produce the actual molecule to study. So I think the structure won't be the rate limiting step. It will be the biochemistry leading up to it.
And in fact, there is a related question regarding this from Edward, and that is, can you predict antibodies on the basis of your structure? I mean, and are we at the time where we can do that? And even if you could predict antibody structures that are best targets for a particular epitope, isn't it a challenge to create a protein with a specific structure? I mean, I think that what he's saying is, can you, from your studies, start to predict antibodies that will neutralize? And can you use those predictions to specify structures somehow?
Yeah, so absolutely, that is the goal. That is the probably 3 to 5 year goal, particularly of my lab, is to really use structural biology, cryo-EM, to solve thousands and thousands of structures, different antigens, different antibodies, And from there, we can then use machine learning approaches to provide a much more predictive approach. So the protein design problem is being solved in many ways by David Baker's seminal work, as well as a lot of the structure-based stabilization that I showed, but really understanding the sort of infinite ways in which antibodies can be made, structured, and recognize antigens is a key missing piece in that design cycle. And I think we can get there if we actually have enough data to feed these machine learning networks and start to iterate from there. And so we're doing a very large campaign in my lab to provide that data to train networks and solve that problem.
And also I got another question that alluded to some of the comments you did. Why stabilization relate to level of protein production? Like, why is so important to produce protein to stabilize the protein first?
Yeah, that's a good question. So as I showed, these proteins undergo large conformational transitions. And once they undergo large conformational transitions, they may be more susceptible to degradation by proteases, for instance. And so the different forms can be degraded. Another thing even earlier is that in protein folding, these are very complicated folding. So over 1,300 amino acids, those polymers have to coalesce into a three-dimensional shape. This can take quite a bit of time, and there's a lot of things that have to go right for that folding to occur. If you use stabilizing mutations, they bias that folding to happen much more quickly. And so I think that that's a key in the ability to fold more quickly, get out on the cell surface, express better, and then once you're on the cell surface, you're stabilized in the immunogenically preferred form, and you're not as susceptible to degradation. So it's a combination of factors that the design really provides for you.
Yeah, another question that came there is, are all viruses using common fusion structures, or each virus has their own fusion machine encoded in their proteins?
Yeah, so conceptually, they're all the same. There's a recognition domain for host cells and then the fusion machinery, but the details are very different, right? So even though we solved the HIV envelope structure and we knew the RSV-F structure, we couldn't predict the coronavirus spike structure. We had to solve that explicitly and determine that experimentally. So I think, again, with AlphaFold is getting better at predicting a lot of these structures, but oftentimes the devil is in the details, and you need to experimentally determine in order to be confident that it's the correct structure.
And the last question, Andrew, that I have given the time is, coming from the is, how do you design these compounds, or how you kind of get, in the case of the transcript, the polymerase, how you come with this is totally empirical, or how do you get to the right compounds?
Yeah, so there's different approaches. There's really a screening approach. So oftentimes companies like Pfizer have 1 million or 10 million compounds that they screen through some phenotypic assay. So they try to do some viral inhibition to down select a certain chemical scaffold, which shows the phenotype that they want. And then from there, structural biology takes over and you look for elaborations. And really it's a game of shape complementarity. So, fitting those shapes and puzzle pieces into the right voids and pockets on the protein. And then really a set of optimization so that you get on target responses. So they recognize viral proteins instead of human proteins. And then, as I showed, adding a modification that results in a covalent inhibitor that really shuts down the protein for good and gives you an advantage. So it's an empirical screening process, down selection, and then design based on shapes and really solving puzzles and taking advantage of what you know about the individual amino acids. Some are sticky, some are greasy, some repel, some attract. And if you can put the right atoms or chemical moieties in the right places, you can have a nice molecule that does exactly what you designed it to do.
And with that, Andrew, I really want to thank you again for a wonderful lecture and to be part of this course, actually. Thank you very, very much.
My pleasure.